I want to welcome Bas. He's an old friend of Coastwise. He's been to talk to us once or twice before, but he has taken events on the shore with us, and they have been superb. So I hope you will view this as a session to prepare you for a field trip in the summer and for some more visits that we will take when we do our surveys to look for molluscs, shells, live or dead animals. They're brilliant things. I'm only just beginning to realize the variety of shells, and that I can see them all times of the year, because the ones that I see on the shoreline, on the strand line, are as important now to me as my lovely nudibranchs are in the summer. So great. Now, Bas says his introduction. He says he's an amateur mollusk researcher working with the Conchological Society. Before he retired, he worked in the Middle East and then in England, first as a biologist working on mammal bones from archaeological sites, especially interested in early history of farming, and then providing scientific advice and managing and commissioning scientific research for English heritage. All of the things he mentions there are potential subjects for a talk, I feel, but that's really interesting background. But what we want to hear from Bas now is about the work of his work as a shell enthusiast, the Conchology Society, what it's like in the South Devon area that he tends to visit more frequently now than up here, and how it compares with what we see and what we can see in the future.
Before I start, just to say thank you for the opportunity to talk to you, and hopefully to encourage more of you to take a greater interest in marine molluscs, because it's an underworked area and one that's of considerable interest. Right, I've already basically covered the patch in that, so let's see whether you can get the next screen properly. Yeah, there we are. Right, there are lots and lots of marine molluscs in British waters, probably somewhere around about 2,000 species, of which something like 800 or 1,000 can be found on our coast. It's a rather bewilderingly large number, and I think that is one of the things that puts some people off them. Essentially, most of them are either bivalves, ones with two shells, or gastropods, and there are several other smaller groups, chitons, tusk shells, and so on. They're both highly interesting and also often rather beautiful, particularly on the smaller detail under the microscope. As the next slide shows some slightly more beautiful bivalves. I should say that they're not completely typical, because they're rather brightly colored ones, and a lot of bivalves are rather dull white. That's the one up top. Top left, Clausinella fasciata, about 1 centimeter across. Across on the right, Veneripis corrugata, rather beautiful one. And then you've got a scallop, Equipectum opercularis there, and a rather invasive mollusk here, Ruditapes philippinarum, came in about 50 years ago from the Philippines and is spreading across the south coast here, bivalves. Typically bivalves in our waters are fairly big, mostly between 1 and 10 centimeters, and they're nearly all filter feeders. In other words, they clean up the water by pumping in dirty water and pumping out clean water, so they perform an enormously useful function in the sea. And most of them are static. They either sit in sand or mud and don't move around much, or they are even cemented to the substrate, though one or two can move around. Some of the scallops are quite mobile. The patterned ones are fascinating, but I'll come back to that later. The next and larger, there are about 100, 200 species that you're likely to find on the shore of bivalve. Moving on to gastropods, there are something like 500 or 600 species that you're likely to find on the shore, and they're much more variable. First of all, in size, they're more variable. Here are some of the bigger ones. Then, of course, you've got a lot of nudibranchs, which have lost their shells, and they're habeas browsers and grazers, which is one of the common adaptations. There are an awful lot of rather specialized feeders. The guy up the top right, for instance, is the Wendell trap, Epitonium clathrus. Feeds mainly on sponges. The guy down left is Yanthena, which it floats around on the sea surface in the open seas. It manufactures a raft of bubbles, hangs stinging cells off it, and traps little jellyfish and eats them. In fact, the stinging cells that it uses are recycled ones that come from the jellyfish that it eats. Then you've got Actian up here, one of the bubble shells. He lives in sand, motors around in sand, and hoofers up detritus and all kinds of things. And rather a pretty thing. He's about 2 centimeters long. Clathrus is about 4 or 5 centimeters. This is the pelican's foot shell, Appareus. It's another one that lives in the sand and burrows around. A rather beautiful shell. The juveniles, of course, don't have this big forked mouth. The juveniles look much more like a straight epitonium. And then here's on the right, bottom right, is the canoe shell, Scaphander, another beautiful shell that again gets up to about 12, 15 centimeters, can be quite big. And that's another one that lives in the sand and burrows around. Those are all the bigger ones. And then if you move down to the smaller ones, here's a range of little ones. Down here is Tricolia pullus, one of the little pheasant shells. He's a grazer. This one is Cerithiopsis, the brown one, who's another specialized one, is sponge eater. These two up here are pyramidelids. They're absolutely tiny. They're only about 2 millimeters, 3 millimeters long, and they're rather specialized parasites living on things like mussels. And then on your left, there are 3 different species of Turbinilla. These are, again, rather specialized feeders. In those 2 cases, we don't know what they eat. This one probably mostly eats Permatoceras, which is that odd annelid worm that lives in those rather triangular shaped calcareous burrows that you find underneath rocks. And this is one of the recurrent themes that a lot of the time we just don't know a great deal about the habits of these animals. In fact, a relatively large number of them have never been found living, which is surprising in this day and age. I meant to say also that almost all the things you've seen so far are found on the coasts of North Devon. The only exception to that, these 2 down here haven't yet been found, but otherwise all the things I've shown you are found off your own coast, not very often in some cases. The problem, one of the big problems about molluscs is the relative lack of public engagement. I don't know how many of you are aware of NBN, which is a wonderful resource in general terms. It acts as a central reservoir of records of what plants and animals have been recorded where in the British Isles. It stands for the National Biodiversity Network. And if you want to look at it on the web, it's well worth it. It's a little difficult to deal with when you start, but it's okay in the long run. And that's its web address. You can see the problem with molluscs when you look at the number of records in different groups. Take a typical well-recorded group like birds. Oyster catchers, for instance, I picked them because they're also on the shore. There are nearly 1 million records on NBN. Turnstones, which are not that common, there's 250,000 records. Avocet even, which is fairly uncommon, 100,000 records. And things like Red Admiral, for instance, there are nearly 500,000 records. About the largest number of records for marine mollusk is the grey top shell, at just under 20,000, which, considering how common it is remarkably low. The common limpet, less than 17,000 records. And that little thing I showed you, Tricholia pullus, probably because of its small size, it's only about 3 millimeters high. There's only about 1,800 records. And the result of this is that quite often when we want to find out more about molluscs and their distributions, we don't have enough information really to understand what's going on, particularly when we're interested in change. I'll illustrate this with a couple of examples. This is taking the comma, which is, as you saw, was well recorded, and its recent spread north. One of the things that NBN lets you do is to split out records from different time periods. On the left here, you've got the records for the northern half of, for the northern part of England and Scotland between 1985 and 1999. There are quite a lot of records nationally, about 30,000. And as you can see, there are just a very small number sitting up in southern Scotland. By 2006 to 2020, which is the same a similar 15-year time period, you've got nearly 200,000 records to play with nationally. But just look at the number in southern Scotland, going right up to Inverness, roughly. It's very obvious that there has been a significant move northwards, presumably as a result of climate change. But when you move to molluscs and look at try to look at the same kind of thing, as you'll see from the next slide, it's much, much less simple because of the lack of records. Looking, for instance, at this animal up here, Phorcus, this is the toothed top shell. I'll show you why in the next slide when we get, look at it now actually. This is a picture of it. It sits in the midshore, it's a grazer, and you can see underneath on the column, none of the other top shells have that. And so if you pick a top shell off the rock and turn it over and look at the columella, if you've got that little lump there, it's Phorcus, and there is only one species in Britain. And so it's very simple to identify. Despite that, between 1985 and 1999, when there were only 500 records of Phorcus nationally, you'll see that most of them are in the southwest, but there's the occasional one right up as far as Chichester Harbour. And in fact, there's one record, that may not be a reliable record, up in the Thames estuary. The prediction at that point was that Phorcus was going to move eastward along the south coast and northwards up the west coast under the influence of climate change. Underneath, you can see the records for 2006 to 2020, again, as in the comma. A much larger number of records nationally, 1,500. But if you look at the pattern along the south coast, it doesn't tell you very much because of the shortage of records in that area. There are a fair number of records in the Solent, more than there were before. But on the other hand, there's a shortage of records in this area. Nothing up here, nothing further east than the Solent, which of course you've got a couple of records here in the earlier period. You look at that, you can't really say that there's any significant change at all over that period. It'd be lovely if people had recorded more, but we simply can't tell. And that's one of the kinds of reasons why we need to do more recording. Moving on to a different example. I mean, one of the reasons I picked this one up is that it is so easy to identify Phorcus that the problem of identification isn't one of the issues when you look at the shortage of records. We've got 3 species of limpet. 3 species, I should say, of limpet with this kind of morphology. There is also a very smooth-shelled one. The commonest one of all, Patella valgata, has colorless tentacles, the pallial tentacles around the edge, and generally a rather sort of dull gray foot or a slightly khaki-colored foot. And the inside is not particular, it's got rather dull rays around. The Patella ulysseponensis, the China limpet, is called because it's got a fairly bright white interior once it's above a certain age. And it's got these little pale white pallial tentacles around the edge. You can see them there. It's got an apricot colored foot. if it's once it's a reasonable size, and is fairly easy to identify. That's, again, all pretty well around all our coasts, just like Patella vulgata. Patella depressa is a southwestern species, and it's very readily identified because of these bright white flecks in the edge of the shell and these very bright white numerous pallial tentacles. And they're further in this case because the mantle is more retracted than it is here. But they're the same, these things are the same as these ones out here. And you can see they're very bright white and they're very startling. The foot also tends to be dark, but that's not absolutely universal. One of the problems is people have been rather muddled about identifying limpets because they try to identify them on too few characters. Another problem is that you have to flip them over to look at them. A lot of people are slightly worried about doing this because they are worried about harming the animal. If you put a blunt knife on the edge of the shell without touching it and then give it a quick tap, it'll come off very easily and not do any damage. If you feel any resistance at that point, stop and don't carry on because that will do damage. I can show you, if we're out on the field together, it's very simple to get them off or get a large proportion of them off and see which limpets you've got. This is the data that we got from NBN. The story again is that they should be spreading east and north. Actually, as you can see, there's absolutely no evidence for this at all. This is the 1985 to 1999 pattern, a lot of them on the south of the coast of the Isle of Wight. This is the 2006 to 20 record. And it's part of the problem is the shortage of records. The other problem, I suspect, is that the records that come are never uniformly distributed. It's a question of the activity of particular individuals. At this point, I know that there were 2 research workers who were particularly active in the Isle of Wight and Solent area, which is why you've got that number of records there. I don't know who was responsible for this lot, but I think it's probably just as common recently and maybe even more common. But look at the shortage of records. That I've been recently up to this area. We got them here at Hurst Castle. We got them at various places along the coast here. We didn't get them inside the Solent, but they didn't get them before. I haven't yet been to the outer shore of the Isle of Wight. Recently, there has been a report of this species even as far as Hastings, but I haven't seen a picture of it. And it's not that easy to identify to somebody who's not very used to them. So I'd like to see a picture. So that, I think, illustrates the problem one has because of the shortage of records, essentially because of the shortage of recorders, and I think some caution and reluctance about a group that's thought of as being difficult. Moving on to intertidal collection methods. For dead shells, it's absolutely simple. You can walk along the tide line, it's enormous fun, just seeing what is there. You can look for shell drift accumulations. These are accumulations of tiny shells and by far the best source of dead small shells. This is a typical example of the kind of place you find them. This is, in fact, on the north end of Briar in the Scilly Isles, and just quite close to the boat landing, in fact. There's this rocky beach, and fairly high up the beach, there's a little patch of dark color. That's mostly little coal fragments and small mussel shells, and in among it, lots of other species. I was lucky enough in it to find a rather rare Wendell trap here, Epitonium trevelyanum, only something like a couple of hundred records nationally. But the more interesting thing is that I got something like 60 species of small gastropod in this accumulation. It's a very clear indication of how many species you find by that route. And a lot of them are very beautiful. Another place to look for dead shells is breaks of slope on the beach. Particularly, you get beaches which are high energy at the top with a steep slope, and then get much flatter at the bottom, and that the kink between the two often accumulates shells. You get boulder groups which accumulate shells, which are quite good sources for looking at the crevices between the boulders. So there are lots of places to look for dead shells, and you get a wide variety of things, particularly if you're prepared to collect things like shell grit and look at it under the microscope. Live shells, of course, give you much more information in some ways. They're more difficult to find, but the problem with dead shells is that you can't be sure how long they've been there. They may sit around on the coastline for certainly tens, possibly hundreds, occasionally thousands of years. And they've moved a bit since they died. I think this has been overstressed. I doubt that many shells you find on the shoreline have moved more than a couple of meters, a couple of kilometers since they died, but they move a bit. Of course, the thing is they get damaged as they go. Live shells are a much better source of information, but as I say, it's more difficult sometimes to find them, particularly shells that live quite close to the bottom of the tide line or even below it. But there are various ways to find live shells, particularly turning boulders over and looking at them, getting samples of weed from rock pools, and doing what's called weed washing. You basically take about a bucket full of weed, put it in, and these are basically mainly you want the red seaweeds, the smaller seaweeds, not the big fucoids, and stick them in fresh water for 12 hours, push them around well, and then take the seaweed out and pour the rest off into a stack of sieves, and you'll find lots of tiny dead sieves, and they give you a much better picture of what there is in that rock pool at the time. Boulder scrubbing, similar kind of technique. Pick up boulders and scrub them over a bucket, and what comes off from the bottom contains a range of molluscs, often very informative. And then again, you can dig and scoop mud and sand, particularly down close to the lower tide line, and put it through a sieve and see what you've got. All fairly simple, low-tech techniques, and all capable of giving you a lot of information. And the equipment is not expensive. If you're talking just about large shells, what you really need is the reference books, and there's lots of websites also that are useful. If you want more information, go to the ConchSoc website, and there's plenty there. It would be hard to spend more than a couple of £100 on good reference books, even if you felt you had to, because there aren't that many around. When you're moving down to the smaller molluscs, so I should also say for the larger ones, you also need things like plastic bags and labels and so on, but that's relatively easy. If you move down to the smaller ones, you first of all will need some sieves, partly to sieve them out, and partly because if you put an unsize graded set of residues under a microscope, it's pretty confusing because the large chunks get in the way of looking at the smaller things. So you want basically a stack of sieves, something like 5mm, 1mm, and 0.3 or something like that, so that you can look at particular size grades to look in more detail at what you've got, rather than being confused by the wide range of sizes. A stack of sieves can cost as little as 50 quid or up to 150-ish, depending on how durable you want them to be and how high precision. Net, you very definitely need a net of some kind, beefed up prawn nets, and a good one will set you back about £50, and that's well worth doing, particularly when you're going to be either dipping in lagoons and so on, or looking at what's in mud or sand. And then the other thing that you would probably need is a decent binocular microscope. You want one that handles the by 10 to by 30, by 10 to by 40 range. And that a good low quality, a good reasonable one, reasonable quality, but low budget end. It costs you about £200. I use one you can probably get for about 100. And they're great fun to look at other groups as well with. And this, so it looks a bit expensive, isn't expensive as compared with, for instance, mountain bikes. I'm very much aware of this from my daughter, who spends literally thousands of pounds on a mountain bike. And the important thing about having looked at shells, having identified them, is to report them. If the information just sits with you, it doesn't do much good. Straight into the NBN Atlas, for instance, or to the ConchSoc, and they put records in, or through things like iRecord. And when you report shells, please don't just report rare ones. Report the whole lot, because otherwise you get a very misleading picture of what's going on, and you don't get reasonable information about the commoner species, as you saw from the limpets and focus that I showed you about earlier. And if you think the species is a rare one, do please either keep the shell itself, what they call a voucher, or keep good photographs, because obviously a rare shell needs to be verified by somebody if there's any doubt about its identification. There's lots of things you can do also to take it further if you want to, but because we know effectively so little about molluscs, I've spent just a little time on that. As one example, there's what they call lagoon cockle. This is a picture of a lagoon cockle. It looks pretty like an ordinary edible cockle, but it's got a much lighter shell. The ribbing goes much further up inside. In an ordinary cockle, the ribbing, you can just see the ribbing on the outside edge, and it tends to have darker patches inside than the edible cockle. DNA differences suggest that it's a completely separate species, and there's no evidence that it interbreeds. We know a certain amount about its distribution, but there's a lot of confusion, and people feel uncertain about identification, because actually when you look at populations, you find a lot of variation and a lot of overlapping. I thought it's a sort of example of what amateurs like myself can do. I thought it'd be interesting to see if relative weight by itself provided a useful guide. And what I did was to measure the weight and the maximum size of cockles in various, cockle shells, I should say, in various populations, and compare them. These blue squares up here with a regression line show what happens when you take an ordinary population of edible cockles. These are ones from Dale Roads in Pembroke. What I've done here is to plot logarithm of the weight against the logarithm of the greatest length. The reason I do that is that if you plot the raw measurements, you get a long curve, and it's very difficult to compare two curves with each other when there's partial overlap. It's much simpler to separate them out if you plot the log of the logs of measurements against each other, because these tend to produce straight, as in these cases, straight lines. It's much simpler. Underneath here, these reddish symbols are two samples of lagoon cockles, one from Pennington, which is in the Solent, the other from Slaughton, which is in Suffolk. And you can see that the weight of these lagoon cockles, the weight of the shells, is much smaller in relation to the length. than in the edible cockles. And that's why the line is displaced downwards, further down the log weight axis. It looks at first sight, and I was very excited when I got this plot to start with, that this was going to be a nice, easy way of telling them apart. But hey ho, the next sample I looked at from a lagoon in Pembroke called called Pickle Ridge, these green triangles, you can see that the regression line here is completely intermediate between the two. So what do we got? Is this evidence of interbreeding? Is it evidence that the method doesn't work at all, or doesn't work usefully? How do we find out? Well, what I'm planning to do is to look at the fleet, which is quite an interesting place anyway. Here in the fleet, the story is that you've got lagoon cockles alone up in this area in the West Fleet, and you've got common edible cockles down in the East Fleet towards the mouth, and that the fleet has a connection with the sea here under a bridge. And that the claim is there is an overlap zone here where you get both species, but they're distinct. So this has never been properly documented. So obviously it's going to be fun to go to the fleet, which is what I'm planning to do fairly soon, to collect cockles from this area, check that they are different and show the same difference in weight against size as we got before. Then look at the overlap zone and see whether what I get there is intermediate specimens, as at Pickle Ridge, or whether you get what are in effect two different populations with different characteristics, therefore broadening the distribution. So what you might expect if you've got intermediates again like this one, you get a line going down like the Pickle Ridge ones, whereas if the two rather broad scatter, which you'd get if you combine the blue ones and the red ones, my personal guess is that these Pickle Ridge specimens are actually just an unusually light ordinary edible cockle, and that the edible cockle could be fairly variable from about here to about here, whereas the lagoon cockle will vary down this way. So you will actually get a separation as long as you don't think that there's a rigid line. But it's not easy, but I think with enough samples, you'll get a clearer picture. That's just one example of what an amateur can do to play with shells and find out more. A question I haven't done anything with, and I don't know how to deal with it, but you might be interested to think about, is why you get such variation in the colour of bivalves. This is a little bivalve that gets up to about a centimetre. It's called Gouldea minima. And why does it have such a wide variation of colour patterns on it? And you saw earlier with the bivalves I showed you, and they're very brightly coloured, the species I was telling you about. And the colour patterning is very variable. What other function could there be in the patterning and the variation? In sand or in mud, where you can't see, they can't see each other. They don't anyway have eyes to do it with. Their predators probably can't get a clear view of them because the water is so muddy. So why are they bothering to be coloured like this? And why was there so much variation? It's a fascinating question, but I don't have an answer at all. I just mentioned it's one of the things we simply don't understand about molluscs. To give you another example, one you probably an amateur couldn't do anything much about, but it's again an interesting question. You've got a very specialized mussel, a thing called Adipicola simpsoni, which lives on whale fall. These are sites where a whale has died and fallen to the floor of the ocean. And you find when a whale does that, that a very well documented sequence of animals move in and deal with the remains and get what food they can from it. Hagfishes, lampreys, this kind of thing, come in first and chew off all the flesh and cartilage that they can get at. And then various other things come in and use and feed on what's left behind. Adipicula is one of these. It's a small mussel, and it moves in, and you get literally thousands of adipicula on a single dead whale at a certain stage in disintegration. But the question is how they get there. But dead whales aren't that common on the bottom of the ocean floor. There are probably, you probably have to go 5 or 10 kilometers, maybe 20 kilometers to get the next dead whale. And you've got an animal here that's not very mobile. Its larva is mobile, but it doesn't have much in the way of time to do things in. And anyway, is up top of the water column right near the surface. In some way, presumably, the larva manages to float around the decaying whale, and then in a very purposive way swims down perhaps a kilometer or two through the water to find the dead whale at the bottom and sit there and prosper and multiply. It's a remarkable story, and one would love to know more about exactly how it happens and why it happens. I hope that some of these examples have illustrated the interest of molluscs, the beauty of molluscs, and the amount we don't know, and how easy it is for even amateurs like you and me to do things to find out more and perhaps help with this. I'd end with a couple of plugs. First of all, I hope you'll consider doing more, and if you do, I hope you'll consider joining the Conchological Society, sending your records in to the HOGSOG or to NBN, and most important, enjoy it. It's enormously enjoyable. One of the fascinating things, for instance, about sorting shell grit under the microscope is that it's just as good as beachcombing, and you don't have to walk in the rain. You're essentially walking along a strand line under your microscope and seeing an enormously magic variety of tiny and rather beautiful molluscs and other things. So thank you very much for listening, and thank you to Paula and to Coastwise North Devon for organising this. And if you've got any questions either now or later, I would be very happy to help. Thank you.